Hello everyone, welcome to the next lecture in our physiology video lecture series. This particular lecture is titled Body Fluids and Kidneys Part 2. In the previous lecture on kidneys, we have already looked into the structure and function of the kidneys, and in this particular lecture, we'll delve into the details how the kidneys process the glomerular filtrate in order to form the urine, the composition of the glomerular filtrate as it travels through various parts of the nephron. And then we'll look into how the kidneys maintain the concentration of various solutes in the extracellular fluid as well as osmolarity of the blood. So let us get started. First off, we will look into the formation of urine by the kidneys. The major forces behind formation of urine by the kidneys is the reabsorption and secretion by the renal tubules. Here we can see a diagrammatic representation of the nephron itself, which is the functional unit of the kidneys. So the kidneys contain about 1 million nephrons, which contribute towards urine formation, but the process across them is the same. As the glomerular filtrate enters the renal tubules, it flows sequentially through successive parts of the tubule. So as we have already seen, blood while passing through the glomerular capillaries are filtered through into the Bowman's capsule and form the glomerular filtrate, which then passes into the whole structure of the nephron and which is known as the renal tubules, and it now glomerular filtrate passes through the proximal tubule, which is the part here, followed by the loop of Henle, which follows like this and has two parts, the descending arm and the ascending arm. Further, the loop of Henle's ascending arm is divided into the thin portion as well as the thick portion. Next, the glomerular filtrate passes through the distal tubules right here, followed by the collecting tubule and finally the collecting duct. Along this course, some substances are secreted, selectively reabsorbed from the tubules back into the blood, whereas others are secreted from the blood into the tubular loop. If you can recall, we have already looked into this phenomenon in which after filtration, the final concentration of the solute in the urine depends on the rate at which it is reabsorbed or secreted while traveling through the renal tubules. Thus, the urine that is formed and all substances in the urine represent some of three basic renal processes, which are the glomerular filtration, tubular reabsorption, and tubular secretion, which can be expressed in this manner, which is urinary excretion rate equal to filtration rate minus the reabsorption rate plus the secretion rate. This is obviously because the filtration rate and secretion rate of a particular solid will determine directly the amount of the solute present in the urine, whereas reabsorption of the solute back into the blood will prevent its presence in the urine, and thus it has an opposite sign to the other. So this is overall how the process of urine formation occurs. Next, we look into the different rates at which the various substances are filtered, reabsorbed, and excreted by the renal tubules. As we can see, substances such as glucose and bicarbonate are almost substances such as glucose and bicarbonate see a almost 100% reabsorption rate, because these are substances that can be reused by the body and used in various parts of the metabolism. Similarly, sodium and chloride are also reabsorbed at a high rate, whereas potassium is reabsorbed at a lower rate. End products such as urea are sparsely reabsorbed at a rate of 50%, whereas end products that can be no longer used by the body, such as creatinine, are completely excreted by the kidneys and no amount is reabsorbed by the renal tubules, and hence we have 100% excretion or 0% reabsorption of creatinine by the kidneys. Filtration can further be defined as the glomerular filtration rate divided by the plasma concentration of a particular solid. The process of glomerular filtration and tubular reabsorption are quantitatively very large relative to urinary excretion of many substances. This means that a small change in the glomerular filtration or tubular reabsorption can potentially cause relatively large change in urinary excretion. This is obviously because these two forces play a huge role in the formation of the urine itself. Contrary to glomerular filtration, which is relatively non-selective, that is any solute that is small enough to pass through the intercellular clefts in the glomerular capillaries will pass into the Bowman's capsule and form the glomerular filtrate. That is, this process is relatively non-selective. Whereas tubular reabsorption is highly selective, and here the kidneys can make sure that particular substances are only reabsorbed while others are completely excreted. As we can see obviously from the table again, where glucose is 100% reabsorbed, whereas creatinine is absolutely not reabsorbed by the renal tubules. Next, we look at the mechanisms for tubular reabsorption, which include both the passive and the active mechanism. In order to reabsorb a substance, it must be transported through the tubular epithelial membrane into the renal interstitial fluid. So, if we see that this is the renal tubule itself on the rightmost of the slide, the substance first needs to pass through the tubular epithelial membrane, which is this layer right here. So, epithelial membrane, epithelial cells are the cells that form the wall of the tubule itself. So, they must first pass through the wall of the tubule and flow into the interstitial space between the cells and the capillary. And after they have been transported to the renal interstitial fluid itself, they will now they must now be transported to the peritubular capillary membrane and back into the blood. So now after reaching the interstitial fluid, they have to pass through the blood vessels or the peritubular capillary membrane to enter the blood flow itself. This transport can occur either via active or via passive mechanisms at both ends of it. So we have further, this transport can be divided into the paracellular path, in which the solutes pass right through pores in the walls themselves, or the transcellular path, in which the solutes pass through the cells themselves. So the cells pick the particular solutes up on their basolateral side and then allow their passage into the interstitial fluid at their luminal side. So solutes can be transported either through the epithelial cells or between either through the epithelial cells themselves or between the cells. Renal tubular cells, like other epithelial cells, are held together by tight junctions. Lateral intercellular spaces lie behind the tight junctions and separate the epithelial cells of the tubules. Solutes can be reabsorbed or secreted across the cells by the way of transcellular pathway or between the cells by moving across the tight junctions, and intracellular spaces by the way of the paracellular pathway. Sodium is a substance that moves through both routes, although most of the sodium is transported through the transcellular pathway via active transport. So coming to active transport, let us look into it. Transport that is coupled directly to an energy source such as hydrolysis of ATP is termed as primary active transport, such as the sodium-potassium ATPase pump in the renal tube. Transport that is coupled indirectly to an active source such as to an ion gradient is referred to as secondary active transport. For example, this includes the reabsorption of glucose via sodium glucose symporter. So in the first case, the ATP is used in order to move sodium across the transcellular pathway opposite to its concentration gradient, and thus it requires ATP, and hence it is termed as primary active transport. In the case of secondary active transport, ATP is still used, but ATP is still used in order to transport glucose, but this involves the co-transport of two things together, that is sodium and glucose. Till both sodium and glucose bind to the transporter protein, the protein will not allow the transport of either of them into the cell itself. And thus, this is a secondary active transport of glucose where directly it is not picked up, but it is picked up along with sodium. And finally, we have water, which is always reabsorbed by osmosis, which is a passive physical mechanism and no energy is required in it. Next, let us look into primary active transport through the tubular membrane. The special importance of primary active transport is that it can move solutes against an electrochemical gradient by utilizing ATP as a source of energy. A good example of primary active transport is the reabsorption of sodium ions across the proximal tubular membrane. So we will look into the transport of sodium ions across the tubular membrane in order to understand how primary active transport in general works in the renal tubules. On the basolateral side of the tubular epithelial cell, so the basolateral side means the side opposite to the side facing to the exterior of the renal tubules, that is towards the interstitium. The cell membrane has an extensive sodium potassium ATP system. ATP is hydrolyzed to transport sodium ions out of the cell into the interstitium and potassium into the cells from the interstitium. So on this side of the tubular membrane, the cells start pumping sodium out of the cells themselves, so into the interstitial fluid, and potassium is taken up by the cells into the tubular epithelial cells. So the sodium is getting out and potassium ions are getting in. This causes a decrease in concentration of sodium ions inside the tubular epithelial cells. This pumping of sodium out of the cells across the basolateral membrane favors the passive diffusion of sodium across the luminal membrane. That is, the luminal membrane is the membrane that is facing toward the tubular lumen or the interior of the renal tubules. So the concentration gradient that is now created due to the lower concentration of sodium inside the cells allows sodium from the tubular lumen to get into the epithelial cells themselves. So this pumping of sodium ions out of the cell across the basolateral membrane favors passive diffusion of sodium across luminal membrane from the tubular membrane lumen into the cell. So essentially, the whole mechanism works. Thus, the whole mechanism works in order to first pump sodium out of the cell on the basolateral side, which causes a decrease in concentration in the cells themselves, and this now promotes the absorption diffusion of sodium from the tubular membrane to tubular lumen into the cells, thus continuing the cycle. The net reabsorption of sodium ions from the tubular lumen back into the blood involves at least 3 steps. The first is sodium diffusion across the luminal membrane into the cells down an electrochemical gradient established by the sodium potassium ATPase pump on the basolateral side of the membrane. Sodium is then transported across the basolateral membrane against an electrochemical gradient by the sodium potassium ATPase pump. So the sodium potassium ATPase pump helps in the pumping of sodium out of the cell and transport of potassium into the cell. Sodium, water, and other substances are reabsorbed from the interstitial fluid into the peritubular capillaries by ultrafiltration, a passive process driven by hydrostatic and colloid osmotic pressure gradients. So once all the solutes and everything is transported into the interstitium, they now travel to the peritubular membrane. They now travel through the peritubular membrane into the peritubular capillaries via the process of ultrafiltration. That is, the molecules which are low in size and molecular weight can directly pass through the intercellular cleft in the peritubular capillary membrane and pass into the blood flow into the peritubular capillaries. Next, we look at the secondary active transport through the tubular membrane. After looking at primary transport, we will look into secondary transport. That is the second example we will looking at. And for that, we will look especially at the sodium glucose co-transport system. Two or more substances interact with a specific membrane protein, which is also known as a carrier protein, and are transported together across the membrane. As one of the substances, sodium, diffuses down its electrochemical gradient, so the amount of sodium in the tubular membrane is high, and that in the epithelial cell is low. As we have seen again and again, sodium concentration in the extracellular fluid is always higher than the intracellular fluid, and thus sodium travels easily via the epithelial membrane of the tubular epithelial cells into the cells themselves. As one of the substances, sodium, diffuses down its electrochemical gradient, the energy released is used to drive another substance, that is glucose, against its electrochemical gradient. Glucose has a different story altogether. Glucose amount or concentration is higher in the intracellular fluid than the extracellular fluid. But since the transport of sodium releases energy enough which can allow glucose transport along with it, this transport of sodium and glucose occurs together against the electrochemical gradient of glucose itself. So if we wanted to transport glucose directly, then we would have to use ATP in the form of energy. But the transport of sodium itself drives the transport of glucose, and since no direct energy source is utilized, this is known as secondary active transport. Another example of secondary active transport is counter transport. The energy liberated from the downhill movement of one of the substances, such as sodium ions, enables the uphill or against the gradient diffusion of a second substance in the opposite direction, such as hydrogen ions. For example, sodium hydrogen counter transport. So this is the second example that we can see right here. As sodium enters into the cell, this energy is utilized by the cell in order to move hydrogen out of the cell. Another active transport mechanism for reabsorption of proteins, which are larger molecules which cannot pass directly through the cell membranes or via the paracellular pathway using the intracellular clefts in between cells, is pinocytosis. In this process of pinocytosis, the protein attaches to the brush border of the luminal membrane. This portion of the membrane then invaginates to the interior of the cell until it is completely pinched off. And a vesicle is formed containing the protein. The protein is then digested into its constituent amino acids, which are reabsorbed through the basolateral membrane into the interstitial fluid. So the protein is directly not reabsorbed or allowed to pass into the blood itself, but first broken down into its amino acids and then utilized by the cell itself. Next, we look into the passive absorption, water reabsorption by osmosis, which is essentially coupled to sodium reabsorption. When solutes are transported out of the tubules, their concentration decreases inside the tubule, while increasing in the renal interstitium, creating a concentration difference that causes osmosis of water from the tubular lumen into the renal interstitium. So as the various solids are passing through the renal tubules, they are actively reabsorbed by the cells. This in order allows the glomerular filtrate to be less concentrated. So less concentrated means that the concentration of water in the renal tubules is higher than it is in the interstitium. And this promotes now the diffusion of water itself from the renal tubules into the interstitium. Some parts of the renal tubule, especially the proximal tubule, are highly permeable to water. Rapid water reabsorption via osmosis occurs through tight junction between the cells. These tight junctions are the intercellular clefts that are present on the borders of various epithelial cells that line the boundary of the renal tubules. In more distal parts of the nephron, beginning in the loop of Henle, the tight junctions become far less permeable to water and solutes, and water reabsorption is greatly decreased. Next, we look into the reabsorption of chloride, urea, and other solutes by passive diffusion. Transport of positively charged sodium ions out of the lumen leaves the inside of the lumen negatively charged compared to the interstitial fluid. So as more and more sodium ions are transported out of the lumen into the interstitium, the glomerular filtrate itself becomes negatively charged. Chloride ions are now able to diffuse passively through the paracellular pathway down the electrochemical gradient. Because there is a lot of negative charge present in the glomerular filtrate, this allows chloride ions to pass through the paracellular pathway passively. The active reabsorption of sodium is closely coupled to the passive reabsorption of chloride by the way of electric potential and a chloride concentration gradient that occurs as the glomerular filtrate passes through the renal tubules. Thus, this is a snapshot of how sodium reabsorptions help in the transport of various other substances across the renal tubules. Sodium reabsorption directly allows the decrease in concentration of sodium reabsorption in the renal tubules directly causes the decrease in concentration of the glomerular filtrate in comparison to the renal interstitium. This allows passive osmosis of water, and thus water is reabsorbed. As water is reabsorbed, the luminal area urea concentration also increases as movement of water out of the renal tubules allows the glomerular filtrate to be further concentrated. This causes two things. First is the luminal chloride concentration to increase, and secondly, it allows the luminal urea concentration to increase as well. Both of these parameters allow the passive urea as well as passive chloride reabsorption. Further, since sodium is reabsorbed from the renal tubules, the glomerular filtrate becomes negatively charged, and the lumen negative potential further allows passive reabsorption of chloride ions down their electrical gradient. Next, we will look into reabsorption and secretion in different parts of the nephron, and we will go through different parts of the nephron one by one and look into how various solutes are reabsorbed or secreted in the different parts. First, we will look into proximal tubular reabsorption. The 65% of the filtered load of sodium and water and a slightly lower percentage of filtered chlorine are reabsorbed in the proximal tubules before the filtrate even reaches the loops of Henle. So this is the proximal tubule that is highlighted here, and the transport of various ions and substances is shown in this diagram here. The proximal tubule cells have a large number of mitochondria to support potent active transport processes. These cells have an extensive brush border on the luminal or apical side of the membrane, that is the side that faces towards the renal tubule itself, and extensive labyrinth of intercellular and basal channels, all of which together provide an extensive membrane surface area. The sodium-potassium ATPase pump provides the major force of reabsorption of sodium and chloride and water throughout the proximal tubule. In the proximal half, sodium is reabsorbed by co-transport along with glucose, amino acids, and other solutes. In the distal half, sodium is reabsorbed mainly with chloride ions due to relatively higher concentration of chloride ions here, which favors diffusion of chloride ions from the tubule lumen to the renal interstitial fluid. So in the first half, we see active co-transport of glucose, amino acids, and other solutes along with sodium. Whereas in the second half, as we have already discussed, sodium reabsorption causes an increase in concentration of various substances in the in the glomerular filtrate, which further allows passive diffusion of chloride ions and other ions. The proximal tubule is also an important site for secretion of organic acids and bases such as bile salts, oxalate, urate, and catecholamines. Many of these substances are the end products of metabolism and must be rapidly removed from the body. The secretion of these substances into the proximal tubule plus filtration into the proximal tubule by the glomerular capillaries and almost total lack of reabsorption by the tubules all combined contribute to the rapid excretion in the urine. In addition to the waste products of metabolism, the kidneys secrete many potentially harmful drugs or toxins directly through the tubular cells into the tubules and rapidly clear these substances from the blood. This is how reabsorption and secretion occurs in the proximal tubules themselves. So next, we move on to the change in concentration along the proximal tubule. So this is a plot which shows us how the concentration of various solutes changes as we move along the proximal tubule itself. As we can see, the substances which are required by the body, such as glucose and amino acids, are rapidly reabsorbed across the proximal tubule so that their concentration almost drops to zero as we reach the end of the proximal tubule. Whereas substances such as creatinine and urea, which are not required by the body and are end products of metabolism, are not at all reabsorbed and their concentration increases. This concentration increase is first due to the secretion of greater amounts of both creatinine and urea from the glomerular capillaries into the proximal tubule itself, as well as further osmosis of water from the renal tubules. As water is removed, obviously the concentration of the various solutes will rise. We can see that sodium and chloride have more or less a constant osmolarity as we move along the proximal tubule. This is because the huge amount of water that is reabsorbed is more or less equal to the amount of sodium and chloride that is also reabsorbed. So since both sodium chloride and water are reabsorbed at the same rate, the concentration of sodium and chloride remain constant throughout the proximal tubule. So although the amount of sodium in the tubular fluid decreases markedly along the proximal tubule, the concentration of sodium and the total osmolarity remains relatively constant because water permeability of the proximal tubules is so great that water reabsorption keeps pace with the sodium reabsorption. Certain organic solutes such as glucose, amino acids, and bicarbonate are much more avidly reabsorbed than water, so their concentration decreases markedly along the length of the proximal tubules. Other organic solutes, as I said, that are less permeable and are not actively reabsorbed, such as creatinine, increase their concentration as they move along the proximal tubule. The total solute concentration, as reflected by the osmolarity, remains essentially the same along the proximal tubule because the extremely high permeability of this part of nephron to water. So if one represents the osmolarity of the whole glomerular filtrate as it passes through the proximal tubules, it remains more or less close to 1, where 1 is the concentration or the osmolarity of glomerular filtrate as it was entering the Bowman's capsule. So it remains more or less constant as we move along the proximal tubule, because as the solutes are reabsorbed, water is also reabsorbed at that rate, so that the entire osmolarity remains constant. So as we are done with the proximal tubules, we now move into solute and water transport in the loop of Henle. The loop of Henle consists of 3 functionally distinct segments: the thin descending segment, the thin ascending segment. So this is the thin descending, this is the thin ascending, and this is the thick ascending segment. The descending part of the thin segment is highly permeable to water and moderately permeable to most solutes, including urea and sodium. About 20% of the filtered water is reabsorbed in the loop of Henle, and almost all of this occurs in the thin descending limb. The ascending limb, including both the thin and the thick portion, is virtually impermeable to water. So this is the first portion that we see that is completely impermeable to water and does not allow transport of water at all. a characteristic which is important for concentrating the urine. The thick segment of the loop of Henle, which begins about halfway up the ascending limb, has thick epithelial cells that have high metabolic activity and are capable of active reabsorption of sodium, chloride, and potassium. So these cells are much larger in size than the normal epithelial cells as we have seen in the thin portion of the loop of Henle, and are also metabolically active, so that they have a lot of ATP that is produced, as well as a lot of mitochondria that can produce these ATPs in order to promote the active transport of various substances such as sodium, chloride, and potassium from the glomerular filtrate. Considerable amounts of other ions such as calcium, bicarbonate, and magnesium are also reabsorbed in the thick ascending loop of Henle. So as the diagram represents, sodium, chloride, potassium, as well as other ions such as calcium, bicarbonate, and magnesium are also reabsorbed, while ions such as hydrogen are secreted into the glomerular filtrate. Next, we move on to the distal tubule, late distal tubule, and the cortical collecting duct, or the cortical collecting tube. The distal tubule is the very first portion of the distal tubule, forms the juxtaglomerular complex that provides feedback control of GFR and blood flow in the same nephron. So, as we have already seen in the previous video, the juxtaglomerular complex, along with the macula densa cells, are responsible for the autoregulation of glomerular filtration rate and blood flow in the nephron. And I would recommend looking back towards the previous video if you are having confusion or difficulty having to recall the juxtaglomerular complex and the autoregulation of GFR. So this is the distal tubule region which is in contact with the juxtaglomerular complex, and thus it is able to help in the regulation and feedback control of GFR. The next part of the distal tubule is highly convoluted and has many of the same reabsorptive characteristics of the thick segment of ascending loop of Henle. That is, it avidly reabsorbs most of the ions, including sodium, potassium, and calcium, but is virtually impermeable to water and urine. So this portion of the distal tubule also is able to actively reabsorb sodium, chloride, calcium, and magnesium, but is virtually impermeable to water, and thus this also helps in concentrating the urine, as water is no longer absorbed. In the late distal tubule and the cortical, next we move on to the late distal tubule and the cortical collecting tubule. The second half of the distal tubule and the subsequent cortical collecting tubule have similar functional characteristics. They are composed of two distinct cell types, the principal cells and the intercalated cells. The principal cells reabsorb sodium and water from the lumen and secrete potassium ions into the lumen. The intercalated cells reabsorb potassium ions and secrete hydrogen ions in the tubular lumen. So essentially the whole late distal tubule and cortical collecting tubule act in order to reabsorb sodium and chloride as well as water from the glomerular filtrate as it passes through it, and allow the secretion of potassium and hydrogen into the glomerular filtrate. Next, we move on to the final part of the nephron, which is the medullary collecting duct. The medullary collecting duct reabsorbs less than 10% of the filtered water and sodium, and thus they are the final site of processing of urine. Special characteristics of this segment are the permeability of medullary collecting duct to water is controlled by the level of ADH or antidiuretic hormone. It is not that this particular portion can reabsorb water and other nutrients with respect to its own special characteristic, but can be regulated with the help of the antidiuretic hormone such as vasopressin. Unlike the cortical collecting tubule, the medullary collecting duct is permeable to urea. Therefore, some of the tubular urea is reabsorbed into the medullary interstitium, helping to raise the osmolarity in this region of the kidneys. Finally, the medullary collecting duct is capable of secreting hydrogen ions against a large concentration gradient, because as we have seen last couple of regions of the nephron have been continuously actively transporting hydrogen into the glomerular filter. And this region, and thus as the urine passes through this region, that is the medullary collecting duct, the concentration of hydrogen is already very high in the urine. Thus, if the medullary, thus in this part, the special characteristic of the medullary collecting duct plays a role, where it can secrete hydrogen ions, even though there is a huge concentration gradient against the secretion of the hydrogen ions into the urine. Whether a solute will become concentrated in the tubular fluid is determined by the relative degree of reabsorption of the solute versus the reabsorption of water. If a greater percentage of water is reabsorbed compared to the reabsorption of the particular substance, the substance becomes more concentrated. If a greater percentage of the solute is reabsorbed, the substance becomes more diluted. Next, we have the changes in average concentration of different substances at different points in the tubular system. As we can see, in the proximal tubule itself, most of the important substances that were filtered, such as glucose, protein, and amino acid that can be reused by the body, are more or less completely reabsorbed, so that their concentration drops down to almost zero. But on the other hand, end products such as creatinine, inulin, they all get more and more secreted into the urine itself with little to no reabsorption, and thus their concentration rises as the urine passes through different portion of the nephron. The case is different for substances such as sodium, chloride, and potassium. First, in the proximal tubule itself, their reabsorption keeps pace with the reabsorption of water, so their concentration remains more or less equal to the glomerular filtrate concentration, which is represented by 1 in the plot. But as we enter the loop of Henle, first there is a huge reabsorption of water, which concentrates these various substances, potassium, sodium, and chloride. But in the second half of the loop of Henle, as we have seen, the water cannot penetrate the thick ascending portion of the loop of Henle, and thus the only sodium, potassium, and chloride are reabsorbed. This reabsorption causes a marked decrease in their concentration as they enter in the distal tubule. In the second half of the distal tubule, again, we know that these substances are reabsorbed rapidly, till finally we enter the collecting tubule and the final parts, where water is reabsorbed in the presence of ADH, which finally again concentrates the particular substances to their final volume in the urine. This is how the entire tubular system works, in where reabsorption and secretion goes hand in hand in order to produce the final concentration of the various solutes in the urine. After we have looked into the various reabsorption and secretion characteristics of various portion of the nephron, we now move into how the tubular reabsorption is controlled, especially by the hormonal system. Here we have a table in which different hormones and their effect on reabsorption is stated. So as you can see, aldosterone has positive reabsorption effects on NaCl and water, and increases potassium secretion. Similarly, angiotensin 2 increases the reabsorption of sodium, chloride, and water, but also increases hydrogen ion secretion. We'll look into various of these as we go along. So first we look into aldosterone and its effect, which is to increase sodium reabsorption and increase potassium secretion. Aldosterone, secreted by the zona glomerulosa cells of the adrenal cortex, is an important regulator of sodium reabsorption and potassium secretion by the renal tubules. The primary site of aldosterone action is on the principal cells of the cortical collecting tube. So as if you just recall, in a couple of slides back, we have looked into how the cortical collecting tubule are made up of the principal cells and the intercalated cells, where the primary action of the principal cells is the absorption of sodium, reabsorption of sodium, and secretion of potassium. The mechanism by which aldosterone increases sodium reabsorption while at the same time increasing potassium secretion is by stimulating the sodium potassium ATPase pump on the basolateral side of the cortical collecting membrane. Aldosterone also increases sodium permeability of the luminal side of the membrane. So as we have seen, the whole reabsorption of sodium acts via two phases. The first is the sodium potassium ATPase pumps that are present on the basolateral side, which allow the transport of sodium out of the epithelial cells into the interstitium, and the diffusion of sodium from the from the tubule lumen into the epithelial cells via the tubular membrane, via the luminal. So aldosterone acts both on both of these characteristics. First, by stimulating the sodium potassium ATPase pump, which allows more pumping of sodium from the cells to the interstitium, and also increasing the sodium permeability on the luminal side, which allows more and more entry of sodium from the renal tubules into the tubular epithelial cells. Next, we move on to angiotensin 2 and how it increases the sodium and water reabsorption. Angiotensin 2 is perhaps the body's most powerful sodium retaining hormone. Angiotensin 2 formation increases in circumstances associated with low blood pressure and or low extracellular fluid volume, such as during a hemorrhage or loss of salt and water from the body fluids. The increased formation of angiotensin 2 helps to return blood pressure and extracellular volume towards normal by increasing sodium and water reabsorption from the renal tubules through three main effects. The first is angiotensin 2 stimulates aldosterone secretion, which in turn increases sodium reabsorption. Angiotensin 2 constricts the efferent arterioles, which has two effects on the peritubular capillary dynamics that raise sodium and water reabsorption. Angiotensin 2 also directly stimulates sodium reabsorption in the proximal tubules, loops of Henle, the distal tubules, and the connecting tubules. So, angiotensin 2 is associated with sodium reabsorption in most of the nephron, and it directly also stimulates the sodium reabsorption in the various parts of the nephron, hence helps in overall increase in sodium and water reabsorption. The antidiuretic hormone increases water reabsorption. The most important renal action of ADH, like vasopressin, is to increase the water permeability of the distal tubule, connecting tubule, and the collecting duct epithelia. In absence of ADH, the permeability of distal tubules and collecting ducts to water is low, causing the kidneys to excrete large amounts of dilute urine. So when the amount of ADH is low, large amounts of water are excreted as well, and thus this creates a dilute urine which has a larger volume. Thus, the action of ADH play a key role in controlling the degree of dilution or constitution of urine. ADH binds to specific V2 receptors in the late distal tubules, collecting tubules, and collecting duct, increasing the formation of cyclic AMP and activating protein kinases. This activated protein kinase cascade leads to increased localization of aquaporine water channels to the luminal membrane and also increases transcription of aquaporine genes. Aquaporine water channels are essential water channels that are responsible for reabsorption of water via the transcellular pathway. That is, the cells themselves become the conduits for water to be reabsorbed and then transported to the interstitium. Next, we look into the effect of the atrial natriuretic peptide and how it decreases sodium and water reabsorption. Specific cells of the cardiac atria, when distended because of plasma volume expansion, secrete a peptide called atrial natriuretic peptide. Increased levels of this peptide in turn inhibit the reabsorption of sodium and water by the renal tubules, especially in the collecting ducts. This decreased sodium and water reabsorption increases urinary excretion, which helps to return blood volume back to normal. Next, we look into parathyroid hormone and how it increases calcium reabsorption. Parathyroid hormone is one of the most important calcium regulating hormones in the body. Its principal action in the kidneys is to increase tubular reabsorption of calcium, especially in the distal tubules and perhaps also in the loops of renal. Parathyroid hormone also has other actions, which include inhibition of phosphate reabsorption by the proximal tubule and stimulation of magnesium reabsorption by the loop of renal. So this was all about the hormonal control of regulation of reabsorption various solutes. Next, we look into regulation of extracellular fluid and osmolarity and sodium concentration by the kidneys as a whole. The kidneys excrete excess water by forming a dilute urine. The normal kidney has a tremendous capacity to vary the relative portions of solutes and water in the urine in response to various challenges. When there is excess water in the body and body fluid osmolarity is reduced, the kidney can excrete urine with an osmolarity as low as 50 milliosmoles per liter, a concentration that is only about 1/6 of the osmolarity of the normal extracellular fluid. Conversely, when there is a deficit of water, extracellular fluid osmolarity is high. The kidney can excrete urine with a concentration of 1200 to 1400 milliosmoles per liter. Thus, as and when required, the kidney can excrete urine with a huge amount of water in it, thus producing a very dilute urine with osmolarity as low as 50 milliosmoles per liter, but excreted at a large volume. And thus, we have removal of water from the body fluids themselves. On the other hand, when there is a deficit of water and water needs to be conserved in the extracellular fluid inside our body, the kidney can excrete urine with a high concentration. That is, the amount of water in the urine is very, very low. And thus, it makes sure that water is retained in the body itself. The antidiuretic hormone also has an effect in controlling the urine concentration. When osmolarity of body fluids increases above normal, that is, the solutes in the body fluids become too concentrated, the posterior pituitary gland secretes more ADH, which increases the permeability of distal tubules and collecting ducts to water. This allows large amounts of water to be reabsorbed and decreases urine volume, but does not markedly alter the rate of renal excretion of solutes. When there is a large excess of water in the body, the kidney can excrete as much as 20 liters per day of dilute urine with a concentration as low as 50 milliosmoles per liter. The kidney performs this impressive feat by continuing to reabsorb solutes while failing to reabsorb large amounts of water in the distal parts of the nephron, including the late distal tubule and the collecting ducts. Thus, in this scenario, the kidney is still able to reabsorb all the solutes that it was during normal functioning, but it does not reabsorb water, and thus it can make sure that a large amount of water is excreted in a form of dilute urine. So in this particular schematic, we have various parameters that are controlled by the kidney as time passes. So if we first look at the osmolarity of the urine itself, we can see that as the large amount of water is drunk, about 1 liter of water is drunk by a person, the urine osmolarity decreases markedly because water concentration in the extracellular fluid now increases. This means that the plasma osmolarity and the extracellular fluid osmolarity can vary and can decrease. But instead, the kidneys control the urine osmolarity and allow a very low concentration of urine to be excreted, which maintains the plasma osmolarity and does not let it vary too much. Similarly, when a large amount of water is drunk, a huge increase in urine flow rate is observed, and thus these also may make sure that the extra water that was intake by the body is excreted in order to not alter the extracellular fluid osmolarity. And finally, the urinary solute excretion rate, that is the amount of solutes that were excreted, does not alter much or at all with even though such huge amounts of urine is being excreted. So the kidney, in regards to the absorption of various solutes, works as well as in this condition as it would have under normal functioning conditions. Next, we move on to tubular fluid. Next, we move on to how the tubular fluid changes its concentration as it flows through various parts of the nephron. Tubular fluid remains isoosmotic in the proximal tubule. As fluid flows through the proximal tubule, solutes and water are reabsorbed in equal proportions, so that little change in osmolarity occurs. That is, the proximal tubule fluid remains isoosmotic through the plasma, with an osmolarity of about 300 milliosmoles per liter. As fluid passes down the descending loop of Henle, water is reabsorbed by osmosis and the tubular fluid reaches equilibrium with the surrounding interstitial fluid of the renal medulla, which is very hypertonic, about 2 to 4 times the osmolarity of the original glomerular filtrate. Therefore, the tubular fluid becomes more concentrated as it flows into the renal medulla. Next, the tubular fluid becomes dilute in the ascending loop of Henle. In the ascending loop of Henle, especially in the thick segment, sodium, potassium, and chloride are avidly reabsorbed. This portion of the tubular segment is impermeable to water even in presence of large amounts of ADH. The tubular fluid becomes more dilute as it flows up the ascending loop of Henle into the early distal tubule, with the osmolarity decreasing progressively to about 100 milliosmoles per liter by the time the fluid enters in the early distal tubular segment. Next, we look into how the tubular fluid in the distal and the collecting tubules is further diluted in the absence of ADH. As the dilute fluid in the early distal tubule passes into a late distal convoluted tubule, cortical collecting duct, and the cortical duct, there is additional reabsorption of sodium chloride. In the absence of ADH, this portion of the tubule is also impermeable to water, and the additional reabsorption of solutes causes tubular fluid to become even more dilute, decreasing its osmolarity to as low as 50 milliosmoles per liter. The failure to reabsorb water and continued reabsorption of solutes lead to a large volume of dilute urine. Next, we look into the kidneys, how the kidneys conserve water by excreting a concentrated urine. The ability of kidney to form urine that is concentrated, more concentrated than the plasma, is essential for survival of mammals that live on land, including humans, where there can be a drought of water that is encountered in any environment. The requirements for excreting a concentrated urine include high ADH levels and high and hyperosmotic renal medulla. A high level of ADH increases the permeability of the distal tubule and the collecting ducts to water, thereby allowing these tubular segments to avidly reabsorb water. As we have seen, ADH allows the reabsorption of water in the distal parts of the nephron, and thus more and more water is reabsorbed, concentrating the urine that is being formed. A high osmolarity of renal medullary interstitial fluid, which provides osmotic gradient necessary for water reabsorption, to occur in the presence of high level of ADH. So if there is a high osmolarity of the renal interstitium, this creates a concentration gradient of water, so that just via the passive osmosis, water can flow into the renal interstitium from the tubular lumen. The renal medullary interstitium surrounding the collecting ducts normally is very hyperosmotic, so that when ADH levels are high, water moves through the tubular membrane by osmosis itself, and no energy is required as it moves into the renal interstitium. From there, it is carried by the vasa recta back into the blood. Next, we look into the countercurrent mechanism which produces a hyperosmotic renal medullary interstitium. Active transport of sodium ions and co-transport of potassium chloride and other ions out of the thick portion of ascending loop of Henle into the medullary interstitium plays an important role in producing the hyperosmotic renal medullary interstitium. So essentially, a lot of solutes are getting reabsorbed from the medullary in the medullary collecting duct region of the nephron, and this large reabsorption of various solutes and their transport into the renal interstitium allows concentration and osmolarity of that region to increase, and this in turn produces a hypoosmotic renal medullary interstitium. Also, the active transport of ions from the collecting ducts into the medullary interstitium also increases the osmolarity of this region, facilitate diffusion of large amounts of urea from the inner medullary collecting ducts into the medullary interstitium, and diffusion of only small amounts of water from the medullary tubules into the medullary interstitium, far less than reabsorption of solutes into the medullary interstitium. That is the selective reabsorptions of only solute and no water at all, increases the osmolarity of the renal medullary interstitium. Next, we move into the control of extracellular fluid osmolarity and the sodium concentration, and how it is achieved via two mechanisms. The first is the osmoreceptor ADH feedback system, and the second is the thirst mechanism. An increase in the extracellular fluid osmolarity, which is more or less similar to the plasma sodium concentration, causes a special nerve cells called osmoreceptor cells located in the anterior hypothalamus part of the brain to shrink. The shrinkage of osmoreceptor cells causes them to fire, sending nerve signals to additional nerve cells in the supraoptic nuclei. This action potential conducted to the posterior pituitary stimulates the release of ADH, which is stored in the secretory granules in the nerve endings. ADH enters the bloodstream and is transported to the kidneys, where it increases the water permeability of the late distal tubules, cortical collecting tubules, and the medullary collecting ducts. The increased water permeability in the distal nephron segments causes increased water reabsorption and excretion of a small volume of concentrated urine. Thus, water is conserved in the body, while sodium and other solutes continue to be excreted in the urine. This causes dilution of the solutes in the extracellular fluid, thereby correcting the initial excessive concentrated extracellular fluid that started the whole feedback loop. This was the osmoreceptor ADH feedback system. The second is the thirst mechanism. The kidneys minimize fluid loss during water deficit through the osmoreceptor ADH feedback system. Adequate fluid intake is necessary to counterbalance whatever fluid loss occurs through sweating and breathing and through the gastrointestinal tract. Fluid intake is regulated by the thirst mechanism together with the osmoreceptor ADH mechanism in order to maintain precise control of the extracellular fluid osmolarity and sodium concentration. The same area along the anteroventral wall of the third ventricle in the brain that promotes ADH release also stimulates thirst. The neurons of the thirst center respond to injections of hypotonic salt solution by stimulating drinking behavior, and thus hence proving that these neurons in the thirst center are responsible for the drinking behavior that is observed. These cells almost certainly function as osmoreceptors to activate the thirst mechanisms in the same way that osmoreceptors stimulate ADH release. Increased osmolarity of the cerebrospinal fluid in the third ventricle has essentially the same effect to promote drinking. Thus, the same neurons that are present in the thirst center, which promote the release of ADH, also promote drinking behavior in order to maintain extracellular fluid osmolarity. Next, we look into the regulation of extracellular fluid and osmolarity via a diagram. So the diagram to the left is essentially the osmoreceptor ADH system that is represented via a flow chart. So first, if there is a decrease in water concentration in the extracellular fluid, that is a water deficit is there. This increases the extracellular osmolarity. Extracellular osmolarity increase stimulates osmoreceptors osmoreceptors, which then allow, after nerve signal conduction, allow the release of ADH. ADH is then secreted into the blood and the plasma, which increases the plasma ADH concentration. After ADH reaches the kidneys, it increases the water permeability in the distal tubules and the collecting ducts. Hence, the water reabsorption is increased. Water reabsorption increase has a direct effect in decreasing excreted water in the urine, and thus this increased water reabsorption helps in dealing with the water deficit that caused the feedback loop to start, and hence the whole extracellular fluid water concentration, volume, and osmolarity is maintained by the osmoreceptor ADH system. Next, we look into the second mechanism, the control of thirst by the osmoreceptors themselves. Increase in thirst is increased by increase in osmolarity, decrease in blood volume, decrease in blood pressure, and increase in angiotensin concentration. It is also promoted by dryness of mouth, as we obviously know. Decrease in thirst reflex is due to decrease in osmolarity, increase in blood volume, increase in blood pressure, decrease in angiotensin 2 concentration, and gastric distension. The stimuli of next we look into the stimuli for thirst. One of the most important is increased extracellular fluid osmolarity, which causes intracellular dehydration in the thirst centers, thereby stimulating the sensation of thirst. Decrease in extracellular fluid volume and arterial pressure also stimulate thirst. A third important stimulus for thirst is angiotensin 2. Dryness of the mouth and the mucous membranes also produce the stimulus for thirst mechanism. Next, we look into integrated responses of osmoreceptor ADH and thirst mechanisms in controlling extracellular fluid osmolarity and sodium concentration. In a healthy person, the osmoreceptor ADH and thirst mechanisms work in parallel. Even with additional challenges such as high salt intake, these feedback systems are able to keep plasma osmolarity reasonably constant. When either the ADH or the thirst mechanism fails, the other ordinarily can still control extracellular osmolarity and sodium concentration with reasonable effectiveness, as long as there is enough daily fluid intake to balance. Thus, the osmoreceptor ADH and the thirst mechanisms work in parallel, and even though if one fails, the other is more or less able to maintain the normal osmolarity of the extracellular fluid and sodium concentration. If both the ADH and the thirst mechanisms fail simultaneously, plasma sodium concentration and osmolarity are very poorly controlled, as is evident from this plot. So, if we plot plasma sodium concentration versus sodium intake daily, we see that when there is normal functioning of both the osmoreceptor ADH and thirst mechanisms, even though there is a large increase in sodium intake, the plasma sodium concentration remains more or less same. The story is completely opposite when ADH and thirst mechanisms are blocked, and thus in that case there is a huge increase in plasma sodium concentration, and thus the extracellular fluid osmolarity and sodium concentration is not at all maintained. Next, we look into the role of angiotensin 2 and aldosterone in controlling extracellular fluid osmolarity and sodium concentration. Both angiotensin 2 and aldosterone play an important role in regulating sodium reabsorption by the renal tubules. Although these hormones increase the amount of sodium in the extracellular fluid, they also increase extracellular fluid volume by increasing reabsorption of water along with sodium. Therefore, angiotensin 2 and aldosterone have little effect on sodium concentration except under extreme conditions. Next, we move into importance of pressure natriuresis and pressure diuresis in maintaining body sodium and fluid balance. One of the most basic and powerful mechanisms for control of blood volume and extracellular fluid volume is the effect of blood pressure on sodium and water excretion, which is known as pressure natriuresis and pressure diuresis. Acute increase in blood pressure of 30 to 50 millimeters of mercury cause a twofold to threefold increase in urinary sodium output, which is effect independent of sympathetic nervous system. So acute increase in blood pressure from 30 to 50 millimeters of mercury directly causes increase in excretion of sodium. This mechanism is not helped along by the sympathetic nervous system and is a function of the kidney itself. With chronic increase in blood pressure, the effectiveness of pressure natriuresis is greatly enhanced because the increased blood pressure also decreases formation of angiotensin 2 and aldosterone. As we can see in this plot right here, acute, which means short-term changes in pressure, increases the sodium output gradually, whereas chronic or long-term changes in pressure maintains a sodium concentration, increases sodium concentration much more effectively and maintains it at a higher level. Pressure natriuresis and diuresis are key components of renal body fluid feedback for regulating blood fluid volumes and arterial pressure. The effect of increased blood pressure to raise urine output is part of a powerful feedback system that operates to maintain balance between fluid intake and output as shown in the figure right here. An increase in fluid intake above the level of urine output causes a temporary accumulation of fluid in the body. So an increase in fluid intake and non-renal fluid loss causes an increase in rate of change of extracellular fluid volume. This causes a parallel increase in blood volume and extracellular fluid volume. So we see due to excess water accumulation or fluid accumulation, there is increase in extracellular fluid volume and blood volume. An increase in blood volume raises mean circulatory filling pressure and pressure gradients for venous routines. So increase in blood volume directly has an effect on heart in the circulatory system, and the heart pumps harder, and this increases the mean circulatory filling pressure along with the venous return. This increased venous return increases the cardiac output, as we have seen via the Starling mechanism, and an increased arterial pressure then causes increased urine output by the way of pressure diuresis. Thus, we have a whole feedback loop which finally increases the renal fluid excretion, and thus the excess accumulation of water that started the whole feedback loop is cured by the final increased excretion of water as in the form of urine. Thus, this balances the increased intake of water. Finally, we look into the distribution of extracellular fluid between the interstitial spaces and the vascular system. Blood volume and extracellular fluid volume are usually controlled in parallel with each other. Ingested fluid initially goes into blood, but it rapidly becomes distributed between the interstitial spaces and the plasma. The principal factors that cause accumulation of fluid in the interstitial spaces include increased capillary hydrostatic pressure, decreased plasma colloid osmotic pressure, increased permeability of the capillaries, and obstruction of the lymphatic vessels. All of these factors can cause accumulation of fluid in the interstitial spaces, which is known as edema. When small amounts of fluid accumulate in the blood as a result of either too much fluid intake or decrease in renal output fluid, about 20 to 30% of it stays in blood and increases blood volume. The remainder is distributed to interstitial spaces. When the extracellular fluid volume rises more than 30 to 50% above normal, almost all additional fluid goes into the interstitial spaces and little remains in the blood. This occurs because once the interstitial fluid pressure rises from its normal negative value to become positive, the tissue interstitial spaces become compliant and a large amount of fluid then pour into the tissues with the interstitial fluid pressure rising much more. As we have seen in previous videos, the interstitial fluid pressure is normally negative, but as more and more fluid gets accumulated, it can rise to positive, which will in turn allow a vicious cycle, which will allow more and more accumulation of fluid in that interstitial space, promoting edema. So if we plot the blood volume versus extracellular fluid volume, a huge increase in extracellular fluid volume, where when blood volume itself remains constant, gives rise to edema as more and more fluid gets accumulated in the interstitial spaces. So that was all we had for today. In this particular video, we have looked into how urine is processed by various parts of the nephron, and as well as how extracellular fluid concentration, osmolarity, and sodium concentration is maintained by various parts, various processes by the kidneys themselves, as well as the osmoregulator center or the thirst center that is present in the brain. These are the references for the part that we have discussed today, which include the chapters 27, 28, and 29 from the textbook of medical physiology by Arthur C. Guyton, 11th edition. I thank you for listening to this video.